Let's have a look at NotebookLM. So I've got a lesson on DNA replication here, and so I've just got, I don't know, 11 slides. So I made that into a PDF, and now I'm going to upload it to NotebookLM. All right, so we can make a study guide. So first of all, here we've just got some questions that we could pose to the class. The study guide, and while that's going, let's have a look at this deep dive function as well, which is amazing. Okay, so here's our study guide. So there's 10 questions, and those 10 questions have answers, which are here. Then there's also essay questions and also glossary of terms. So that's quite a nice sort of study guide. As well as that, we're making a conversation, basically making like a podcast between two people. They're going to be talking through the content in a really engaging way. So that takes a little bit longer, of course, to put that together. All right, so we have got this audio now, so let's have a listen to that.
Ever wonder how scientists pinpoint a gene linked to something like, say, a rare disease? It's amazing.
It all boils down to DNA replication, really. It's fundamental cells passing on genetic info when they divide, right?
Right. And your notes have me thinking this DNA replication thing is way more complex than just unzip and copy.
Way more. Think intricate dance of enzymes and molecules. Every step choreographed perfectly for the whole thing to work. It's elegant, really.
Okay, color me intrigued. Break it down for me. Why is DNA replication so important?
Imagine this. A cell divides, has to give each daughter cell a full instruction manual, right?
Yeah.
The DNA. Replication makes sure each one gets the complete set.
Like each twin getting the full cookbook, not just half the recipes. Love it.
Exactly. And the copying process.
Yeah.
Super cool. Semi-conservative replication.
Semi-conservative. Sounds almost, I don't know, fashionable for molecules.
Think of it like this. You have your original DNA, the master plan. It unwinds. Each strand becomes a template, and a new matching strand is made alongside. So each new DNA has one old, one brand new strand, hence semi-conservative.
Using the old blueprint to make a new one, keeping part of the original intact. Clever.
Exactly. Now, want to get a little deeper. 4 main stages: initiation, separation, elongation, and termination.
A 4-step cellular dance. Your notes had me hooked on separation, though. That's where the real action is, right? Something about helicase.
Ah, yes, helicase. Our star player. Picture DNA like a zipper jacket. Helicase is the zipper pull, unwinding the 2 strands of the double helix, unzipping to reveal that genetic code inside.
So it breaks those bonds holding the DNA strands together, making the code accessible for copying. Then what?
Enter DNA polymerase, the master builder. It adds nucleotides, the DNA building blocks, to those new strands being formed, creates a perfect match to the original template.
Like a microscopic Lego expert, snapping those nucleotides into place according to the original kind. Makes sense. But there's a catch, isn't there? Something about which direction it builds in.
You're sharp. DNA polymerase, it's a bit picky, see? Only adds nucleotides in one direction, 5 to the 3 fit end of the DNA strand. Like, building a wall can only add bricks to the top one way.
Leading to the whole leading and lagging strand thing, right? This is where I usually get tripped up.
No worries. Think of the leading strand like smooth sailing. It's synthesized continuously, following the replication fork where the DNA unwinds. Like paving a road straight ahead, uninterrupted flow of new DNA.
Got it. But the lagging strand, why is it lagging?
Because it has to be built in reverse, which is tough for our one-way builder, DNA polymerase. So instead of one smooth process, it's done in short bursts, little chunks called Okazaki fragments.
Help me visualize this. Okazaki fragments.
Imagine building that brick wall, but backwards. You have to do it in sections, right? Lay a few bricks, connect them, then another section. That's essentially the lagging strand.
So these fragments are like DNA chunks built in reverse that need to be joined up afterwards.
You got it, and the cell has it covered. Another enzyme, DNA ligase, swoops in its R molecular glue, joins those Okazaki fragments, make sure we end up with a complete, continuous DNA strand.
We've got helicase unzipping, polymerase building, ligase gluing, a whole construction crew in our cells. But why should we care about this tiny team's hard work?
This is happening in every cell, constantly. And it's not flawless. Sometimes mistakes happen during replication, mutations in the code.
Mutations, those are always bad news, right?
Not so fast. They can be the engine of evolution. Imagine a helpful mutation that helps an organism thrive in a changing world. On the flip side, mutations can also lead to diseases, like cancer. It's a two-sided coin.
Wow, never thought of it like that. Always assumed mutations were the villain, but they're both hero and villain.
And context is key, right?
Yeah.
They can drive positive change, but also disrupt how cells function.
Okay, so how will I use this in the classroom? Normally what I do is I make my PowerPoint slides, and then I make a video of those PowerPoint slides, and I make that available for my students for pre-learning. Now, I could use these questions to test their understanding of the pre-learning, so it's more active pre-learning. That would be ideal. Students could then check their answers following that as well. When they come into class, we could do these warm-up questions, which one or two of these. They could take this glossary of key terms and stick that in their books or in their OneNote for their own independent study, or I could put that in there as well. I might use these questions here as our warm-up questions at the beginning of the next lesson. How would I use this podcast deep dive? Not sure yet. I think I need to wait and see what the students think of it. I kind of like it. It doesn't cover all of the information, but I really like the analogies, and I really like the relevance, and I really like how easy it is to listen to, just like a podcast.